In September of 2019, a Harvard geneticist stood on a stage in Boston and told an audience that the first person who will live to 150 years old has already been born. The room did not laugh. David Sinclair has spent 3 decades decoding the machinery of aging, and his book Lifespan argues the most destabilizing claim in modern biology, that aging is not an inevitability but a disease with an information theory, a treatment hypothesis, and a small pharmacy of molecules already sitting in trials. This is not a book about wellness. It is a manifesto for a war on aging itself. In this video, we walk through the information theory of aging, the hallmark damage list, the Sirtuin story that made Sinclair famous, the molecules his lab studies, the lifestyle levers he says activate the same pathways, and the honest skepticism that trails the whole field like a shadow. One by one, let us get into it. Start with the reframe, because everything depends on it. Medicine classifies aging as a risk factor, not a disease, which is why no drug has ever been approved to treat it, because you cannot treat what is not diagnosed. The World Health Organization only recently began discussing an aging-related classification extension, and the regulators remain bound by a system built one disease at a time. Sinclair wants aging reclassified as a medical condition with a treatable mechanism, and his information theory supplies the mechanism. In his framing, the genome is the hardware, the epigenome is the software, and aging is what happens when the software degrades. The DNA itself mostly stays intact, but the epigenetic marks, the annotations that tell each cell which genes to read and which to ignore, drift over time like a scratched compact disc. A skin cell starts reading liver instructions. A nerve cell forgets its address. The cell does not die, it loses its identity, and a body made of confused cells exhibits every symptom we call aging. On this theory, wrinkles and frailty and failing memory are not the wearing out of parts. They are noise accumulating in a control system, and noise can, in principle, be corrected. The theory also explains a puzzle that pure damage models cannot: why identical twins diverge with age, why cells with the same DNA and the same birth date end up aging at different speeds, because information degrades stochastically, copy by copy, division by division, and two systems accumulating random noise drift apart even from identical starting points. The field supports the framing with the hallmark list, the 9 or so categories of damage that accumulate with age, and they read like an engine diagnostics report: genomic instability, the mutations accumulating in DNA, telomere shortening, the fraying protective caps we will meet properly in another video, epigenetic alteration, the software drift at the center of the theory, loss of protein control, the misfolded molecules that clump in aging brains and muscles, mitochondrial decline, the power stations losing efficiency, cellular senescence, the zombie cells that refuse to die and poison their neighborhoods with inflammatory signals, stem cell exhaustion, the repair crews running out, altered communication, the chemical signals between cells degrading, and a dysregulated nutrient sensing network, the growth circuits that never receive the signal to switch from growing to repairing. The list was assembled by a consensus paper the field now treats as its constitution, and its power is diagnostic, because it converts the vague complaint of getting old into a checklist of specific, measurable breakdowns, each one a target. Every longevity intervention ever discovered, from calorie restriction to exercise to rapamycin, works by acting on one or more of these hallmarks, which is why the list functions as the map of the entire field. It also explains the multiplying strategy of modern longevity companies, one attacking senescent cells, another telomeres, another mitochondria, another the epigenome itself, because a map with 9 territories invites 9 armies. Now the Sirtuin story, because it made Sinclair career and controversy in equal measure. Sirtuins are a family of proteins that act like genome supervisors, traveling along the DNA, repairing breaks, and maintaining the epigenetic annotations. The founding observation came from yeast, where extra copies of the Sirtuin gene extended lifespan by up to 30%, and the molecule that activated the protein turned out to be nicotinamide adenine dinucleotide, NAD for short, a coenzyme every cell uses in energy metabolism. The work traced back to a line of curiosity research that began with a screen for genes that silence other genes. Pure basic science with no medical application in sight, a detail Sinclair tells with affection because it is the argument for funding curiosity at all. The catch is that NAD levels fall with age by as much as half between youth and old age, which means the supervisors run out of fuel exactly when the genome needs the most supervision. Breaks accumulate, Repairs fall behind, the annotations slip, and the noise compounds. Mouse experiments added the drama, animals engineered for extra sirtuin activity or given NAD precursors showed better mitochondrial function, better muscle, better metabolism readings, the picture of mice aging differently than their littermates. The Sinclair hypothesis in one sentence is that falling NAD silences the sirtuins, and silenced sirtuins are the engine of epigenetic drift. So the obvious question became, can you refill the tank? The candidate molecules are the pharmacology section of the book. NMN nicotinamide mononucleotide is the direct precursor the body converts into NAD, and Sinclair himself takes a gram every morning, a disclosure that doubles as an endorsement and a red flag depending on who you ask. NR nicotinamide riboside is a related precursor sold widely as a supplement, with a slightly better studied safety file and a slightly different conversion path. Resveratrol, the red wine molecule, entered the story as a sirtuin activator and vaulted into fame on 60 Minutes and in red wine marketing, before independent labs struggled to reproduce the original claims at the doses humans actually absorb, and before the field quietly moved on from wine as a delivery system. Metformin, the diabetes drug, is the longevity molecule with the strongest population data, decades of diabetics on the drug showing surprising mortality advantages, and the large clinical trial designed to test it against aging, the TAME trial, is the field attempt to force regulators to treat aging as an endpoint. And rapamycin, the immune suppressant discovered in the soil of Easter Island, remains the molecule with the most consistent lifespan extension results in mice, extending life by roughly 10 to 30% across strains and sexes, a result so robust that longevity researchers argue mostly about its mechanism and its dosing schedules rather than whether it works in the mouse. Sinclair takes NMN and metformin, exercises, skips breakfast, and describes his biological age testing in the 40s, while his chronological age passed 50. Read that sentence twice, once as a scientist describing his protocol, and once as a consumer asking who profits when the protocol becomes a product, because both readings are required equipment for this field. The lifestyle chapter is where the book touches your kitchen, and the mechanism is elegant. Calorie restriction, the one intervention that extends lifespan in every species tested from yeast to monkeys, works not through weight loss but through stress, the mild hormetic stress of scarcity, which flips the cell from growth mode into repair mode, raises NAD, and activates the sirtuins. The discovery traces back to the 1930s, when nutrition researchers noticed that rats fed Spartan diets lived dramatically longer than their well-fed cage mates. A finding so uncomfortable, it was essentially shelved for decades. Exercise does the same through a different door, depleting energy and forcing the adaptation machinery awake, and the book treats it as the most reliable NAD booster available without a prescription. Cold and heat exposure stress the system similarly, saunas and cold plunges sitting in the same hormetic family. Even the humble act of feeling hunger, Sinclair argues, is a signal, the body announcing that maintenance is now the priority, which is why he practices a morning fast, and why the intermittent fasting movement borrowed heavily from this literature. The enemy, in his telling, is the comfortable, overfed life that never sends the repair signal at all. 3 meals plus snacks, climate control, zero hunger, zero cold, zero effort, and a biology that never once flips into the maintenance mode it evolved to depend on. The irony is sharp. The organisms built for scarcity are dying of comfort, and the cure the book proposes is a deliberate, measured reintroduction of the hardship the species was designed to survive. Then there is the reprogramming frontier, the most science fiction chapter of the book, and the one that may matter most. Japanese researcher Shinya Yamanaka identified 4 transcription factors that can take a fully differentiated adult cell and rewind it back to a stem cell, erasing its age along with its identity, work that won the Nobel Prize and rewrote the rules of developmental biology in a single paper. Sinclair Lab performed the experiment that anchors the information theory, restoring vision in aged mice by briefly switching on those factors in damaged optic nerve cells, letting the cells remember how to be young without forgetting what kind of cell they were, and the animals regained sight that age had taken. The result is that aging, in a tissue, in a mammal, was partially reversed for the first time. The usual caveats apply with full force. A mouse eye is small, the exposure was brief, cancer risk from reprogramming factors is the dragon guarding this treasure, and uncontrolled cellular reprogramming is the textbook definition of a tumor. The gap between a mouse eye and a human body remains enormous. But the principle is now in play, that age is information, and information can be reset, and entire biotech pipelines are now built on the attempt to reset it safely. Sinclair closes the chapter with the image of a reprogramming dial rather than a switch, the search for partial, controlled rewinds that restore function without erasing identity, and that search has become one of the most crowded races in modern biotechnology. Now the skepticism, because a manifesto this confident deserves a defense attorney, and the field has one. The resveratrol saga damaged the reputation of Sirtuin science when key papers were retracted and replication attempts faltered, a story that began with wine glasses and ended in scientific disgrace for a research partner of the era, and the aftershocks still shape how skeptics read everything Sinclair publishes. Critics, some of them eminent, contend that the entire NAD supplementation market runs ahead of any demonstrated human benefit, that blood levels of NAD rising tells you nothing about lifespan until trials say so, and that mice are not small humans no matter how often the graphs rhyme. NMN has no completed human longevity trial. Sinclair financial interests in the supplement industry are disclosed and still uncomfortable. The mouse lifespan studies behind several claims have sample sizes that would embarrass a cardiology trial, and the field has a long memory of interventions that cured every rodent and did nothing for a single patient. And the deepest objection is simply that aging may be too complex, too many intertwined systems failing at once, for any single lever or theory, and that information is a metaphor doing the work of a mechanism. There is also the fairness objection the book takes seriously, that radical life extension arriving first for the wealthy would split the species into the long-lived and the short-lived along the fault lines of money, a question the final chapters wrestle without fully pinning down. These objections do not sink the book, and Sinclair answers several of them directly, but any honest summary hands them the floor. Where does this leave you? Between two certainties that are less certain than they look. The first is that aging is fate, written in stone, untouchable, and the book demolishes that, because the biology of aging is now a mapped, manipulable process, and no serious scientist in the field still believes it is untouchable. The second is that the treatments are ready, available, and waiting in a supplement bottle, and the book does not prove that, because the trials have not finished, and a man who takes a gram of NMN each morning is running an experiment with a sample size of 1. What is left is the honest middle, where the safe levers are old ones, exercise, fasting intervals, not smoking, sleeping, the mild stresses that activate the repair pathways we have mapped, and where the molecules stay interesting, unproven, and advancing. Sinclair closed his manifesto with the prediction that the person who lives to 150 is already alive, possibly watching something like this video. Whether that proves true is a question for a trial still being designed. But the machinery he spent a life mapping is real, the noise theory has survived its first tests, and the war on aging is no longer a metaphor. It has labs, budgets, molecules, and a map. The rest, as the field likes to say, is time.